Please keep the plates clean and handle with care. And I will be taking it home when it doesn't stay. Hi, what's your name?
Christopher.
Christopher.
Yeah.
Your job today is to make sure that I don't leave this room without that book.
Yeah.
Okay. Good.
Your job is to make sure.
Oh, okay. I have no problem with team effort. We're waiting for Saman. Who else? Where's Elec these days?
Elec is in Yale.
Is where?
Yale.
Okay. Well, Mary, I saw. Maria's on nights.
I don't know where any of
That's St. Chris.
St. Chris. Someone who's studying in St. Chris.
Who's in clinic for Van and anyone else?
Someone.
No problem.
I think that's still playing. Let me just check them.
Can you just touch them and ask them if they're on their way? Who's your neuro resident on consults right now?
Steve.
Okay, good. So you're here. And who's on inpatient?
Colin and Mary.
Round stopped by 11:45.
Yeah, that's good.
Would you text Mary then and ask her where she is, please? I'm actually starting to hear tremors of administration starting to think about expanding the neuroscience unit. Probably not in your lifetime and not in my lifetime for sure, but they're starting to. I think I have driven home the point that the unit is too small. I think they have heard it. After 6 years. And the other thing that they've heard is that the combination of a residency, which is too small, and a unit, which is too small, without a neuro ICU is a problem. And I think they're getting that. I mean,
The solution, that's going to be another.
solution,
The first step is recognizing a problem.
that's right. You cannot solve a problem until it has been defined.
Contemplation.
Did either Saman or Havan like that yet? And
No.
That was something.
I'm probably going to pick up a couple weeks of inpatient attending from Ananda at some point. I don't know, maybe a week in November. I'm looking at see if I can do that. I feel the need to do it. I want to. I miss it. And Ananda has been on continuously nonstop since she got here. And she could use a break. So.
Yeah, she's done so many weeks and weekends.
They're on their way. That look like cafeteria. It's a little concerned.
Yeah, it looks like it's curdled.
Yeah, or stuff. It's mango and pickles and strawberry. Mango. Mango and strawberries and pretty. My husband's on a diet. He makes me an extra one. And I go eat extra food in that.
So you got it, so you get it.
I'm on a double diet.
What do I mean? 2 diets?
Did you get from cafeteria?
Oh, yeah.
Where's someone?
It's still open.
Did that young lady with the amyloid
Yep. Monday?
Well, she was pronounced on Sunday, but
Sunday.
they didn't exhibit her till like Wednesday. Family was didn't want us to do anything.
Oh, she got the same thing.
We had like, I think legal was involved because I think there's a policy of exhibiting after 24 hours. That's what I was told. We waited more than 48.
Havan finished summer.
Pavan should be wrapping up. Dr. Pine was good.
Okay, all right, then I'm gonna start, I think. Or whatever it is, we're gonna run out of time. And turn off the air conditioner, please. Yeah, turn it off. I just like to cool the room and then. So, yeah. I would say that let me put it this way. When I prepare for this lecture, I am always blown away by this complexity of the central nervous system. And I don't know of any other lecture that makes me stand more in awe of the brain than this particular lecture. There's a primary book on electron microscopy that can be circulated. Again, I say it's an old book, it's a rare book. Please be careful with the plates. Most of the pictures that almost all the pictures that are there are going to be a part of today's lecture. So today is about the synapse. And we're going to cover vesicle formation, the structure of the synapse, the process of exocytosis, and vesicle recycling. And just very briefly, talk about a general introduction to neurotransmitters, which will be the topic for the second half of the year. So a synapse is a communication between two electrically excitable cells. That's what all synapses share that, and that's about the only thing that all synapses share. After that, you spin off variations that are almost infinite in variability. The synapse, we always think, when you think of a synapse, you're thinking in terms of, most of the time, you're thinking in terms of a chemical synapse, but there are also pure electrical synapses, which we'll discuss briefly. In the central nervous system, you will find receptor cell to neuron, neuron to neuron, and then in the peripheral nervous system, also neuron to muscle, and the peripheral nervous system receptor to neuron. There probably are also glia to neuron, neuron to glia, glia to glia, and every imaginable combination that you can think of. Here's where the complexity starts. Look at this, the number of synapses in each of your brain, okay? So if you assume roughly that there are 100 billion neurons in the human brain, which is approximately right, and that there are roughly 500 synapses per neuron in the brain, which is roughly right, then you calculate that there are at least 50 trillion synapses in your brain, which, to put that number in perspective, is equal to the number of stars in the known universe. Okay? That's how many synapses there are in your brain. Okay? Now, the electrical synapse, very briefly, is an extremely narrow synaptic cleft, okay? Your ruler, remember, is the bilayer, is the membrane bilayer, which is 7 nanometers. So this is no more than the thickness of a half of the bilayer. In an electrical synapse, there are no vesicles, there is no fusion, there are no receptors. What there are gap junctions, and that's it, okay? The advantage of an electrical synapse is that it's very fast. you have a time frame of 100 microseconds, okay? So this is very fast. And I put on here the crayfish. If you just think of when the crayfish encounters danger, it just flicks its tail and it's gone, okay? That flick is nearly instantaneous as far as you can tell on any, and that's because the synapses are electrical. It doesn't need to think. It encountered it, it's gone, and the flipper goes forcefully. Cortical in the human brain and the mammalian human brain, there are cortical interneurons and astrocytes, which basically use electrical synapses to create syncytia, so that they fire nearly in unison. That doesn't happen very frequently, but sometimes you just want to stop something that's going on. This is a great way to do it. Just connect a bunch of inhibitory neurons. When one of them fires, they all fire, and that's the end of it. The system is shut. If you look at the structure of the electrical synapse, it is, again, an extremely narrow synaptic cleft, and really all that you have in them are opposing gap junction and ion channels, and that's basically what they are, and that's well illustrated here in this three-dimensional drawing. And that's it. That's all I'm going to say. From now on, if I say synapse, I mean a chemical synapse. But remember that there are electrical synapses, including in our own brain. When you talk now, the structure of the chemical synapse is it has a width, the gap, the synaptic cleft is about 20 nanometers. Again, your ruler is a bilayer, it's 7 nanometers. So this is at least 3 times the size of a bilayer. You release the neurotransmitter into the synaptic cleft. The neurotransmitters then diffuse mostly through the synaptic cleft and encounter the postsynaptic neuron or cell, where they then interact with a receptor of some sort. And so just think for a second, again, now here comes another level of complexity. You think, so you've got 50 trillion of these going in your head right now, okay? Think about the machinery that's involved in this process of having a synaptic vesicle in a terminal opposed to a receptor plate of a receptor cell, that with the right timing and not with the wrong timing. So through some summation of electrical signaling into the presynaptic neuron, that it decides to take this bundle of prepackaged neurotransmitter and release it into the synaptic cleft, and then take it back up again, which we're going to have to get to. All of this has got to go on. 50 trillion synapses. And if we started to think about how often these things fire, so these things are firing tens to 100 times a second, okay? That's what's going on in your head right now, okay? In a coordinated way. And I think it's worth thinking about how a system like this could evolve. As most of you know, if you've been with me through other lecture series, it's like evolution is an amazing thing. And you can't jump to a system like this. The amount of energy, 88% of the energy of our brain consumption, which, as you know, is the Achilles heel of the brain. We get 20% of the cardiac output to 2% of the mass of the body. It's running at 39 degrees because it's so metabolically active. 90% of the energy is being used for the synapse. So how do you get there? Why would you ever invent a system like this, okay, at that kind of energy expenditure? Okay. That's really the question that I think you have to think about that. All right. Neurotransmitters, back at the more mundane level, back to structure and function here, is neurotransmitters are typically small molecules, meaning a few hundred Daltons, so 200 to 300 molecular weight, but they can also be neuropeptides, and neuropeptides are usually in the 5,000 to 10,000 Dalton range. At the beginning, so I was, at the beginning of my PhD was the beginning of synapse research, when De Robertis in Argentina and Whitaker at the NIH discovered that you could isolate synaptic vesicles, right, synaptic terminals, very simply out of rat brain. You homogenize them, put them in a sucrose gradient, spin the thing at 40,000 and pour it off. And look, and you have just all the synaptic structures that you could ever want to do research on. And if you wanted to, you could break those and do synaptic vesicles, which was actually where I started my PhD. Okay? It was simple. There was nothing hard about it. And that's the beginning, is 1969 is when they, both of them, reported the ability to isolate these. When they were initially reported, it was thought that you had one neuron, one neurotransmitter, as everybody said. Quickly, we learned that that is not strictly true, because although the one small neurotransmitter is true, you wouldn't find a synaptic terminal that had acetylcholine and norepinephrine in it. That doesn't happen. But you did have peptide Y, okay, or that sort of thing, along with the small neurotransmitter. And that didn't count ATP. Everybody, there's ATP that is complex with most of the neurotransmitters. Sometimes you'll have a complex of each neurotransmitter will have 4 ATPs bound to it. There's a 4-to-1 complex that forms, and the ATP is released with the neurotransmitter. I think most of us assumed that the ATP was there in order to do what ATP does, which is to provide energy for the neurotransmitter to do its function. I think that was an overly naive and simplistic approach, and I think we now recognize that the ATP is also functioning as a neurotransmitter through purinergic channels as well as supplying energy. The time course of a typical synapse is 1 millisecond. Now, the time course of the electrical synapse was 1/10 of a millisecond. So this is 10 times slower than an electrical synapse, okay? And what's the advantage? Where do you get, why do all this? And the answer is you have extreme control over this situation. This is what this is about. This is tailored to what we do, which is think, okay? And here's a schematic diagram then of what we have to solve in the rest of this lecture. And that is, how do you get from these synaptic vesicles, cause them to fuse with a presynaptic bilayer, release the neurotransmitter, activate the neuroreceptor, and then we're not going to be dealing with all of this. This, or each of you or faculty will be dealing with this after January 1. Now, the synaptic bouton or button from the French term really comes from light microscopy, when people were finally getting to 100x and looking at the nervous system and getting the right stains in place, and they realized that axon terminals have these little boutons, buttons at the end of them. And then this book, and the people who wrote this book, Peters, Chan, and Palade, then began to do this electron microscopy, and they suddenly made us realize what really was going on in the brain. The fact that you could isolate them wasn't enough. We had no idea about the complexity of the interconnections until this book was published. And one of the things that you will notice in this book, and when you isolate terminals, is the mitochondrial content is through the roof. This is your energy intensive section of the brain. It's not the axon, it's not the myelin, it's not the cell body, okay? This is where the energy is being consumed. If you're consuming energy, you're going to have tons of mitochondria, and every picture you'll see, just the boutons are filled with mitochondria. And then over the years, we have realized that, again, increasing the complexity is that your average synaptic terminal is using about 1,000 proteins, 1,000 proteins to accomplish this work, okay? We're going to be simplifying. We're going to look at 15, 20 proteins and not in a lot of detail, okay? The electrical synapse or the gap junction would have been worth a 1-hour lecture. So I'm going to show you a lot of pictures. They are the pictures from this book. You can look at them here. I brought the book because the detail is greater than in my photographs, although these were photographed professionally, all of them. Look at what's going on here. Here are synaptic vesicles, which are themselves bilayers, mitochondria, okay? Here's a cleft. There's a dendrite, a cleft. Here is a synaptic density. We're going to be looking at a lot of pictures of synaptic density. Here is an extra piece of membrane going on. Remember, this is electron microscopy, and therefore is prone, like all other experimental techniques, is prone to artifact. And it took us decades, but I mean decades, to figure out what could be eliminated, in which case it was artifact, or what could not be eliminated, in which case it was accepted as the truth. Most of these fixations are done by perfusing the rat intracardiac with you. The rat is still alive. You anesthetize them. You then put a catheter directly into their aorta, okay, right through the heart into the aorta, and then instantaneously perfuse them with 4% glutaraldehyde. And they just go like this. And their body is rigid, okay? And then you take the brain out and you fix it with osmium tetroxide, which just is a intense fixative. And then what you end up is a plastic block, which is, you could take a hammer and smack this thing and not be able to break the plastic that it's in. And then you cut this at 60 angstrom thickness with silver and gold tone. And I used to do this. It's the whole technique is unbelievable. Okay, here you see the synaptic terminal. Here's the synaptic vesicles have accumulated to the active zone of the synapse. You might be able to imagine that there are horizontal bars within the synapse here. I wish I could blow these up for you, but they are in the pictures there. You can see them. You can get a sense that they're here on this picture. Those are real. Those are there. Those are part of what make up the synaptic cleft. Here is a single synaptic, a single dendritic, what do you call them? Also, just dendritic spine, okay? This particular dendritic spine has, in this two-dimensional picture, 9 synapses on it, okay? Want to think a little bit about the complexity of decision-making, okay? This is a good place to start. That you have each of your big neurons up in the cerebral cortex has a huge ramifying dendritic tree, and throughout this dendritic tree are dendritic spines, and on these dendritic spines you're having innumerable synapses that are feeding information into this thing, and all of this being summated, everybody having its say in the decisions that we make in the activation of this postsynaptic neuron. It gets way worse than that. Here's a dendrite, here's a synaptic terminal, here's what you'd expect. It looks very ordinary, right? You've got, okay, synaptic vesicles coming to an active zone, activate the dendrite. Oops, you have a dendroaxonal synapse over here. Now the dendrite is doing the work and is exciting the presynaptic, which is presynaptic now. Is the dendrite presynaptic or postsynaptic? It's there. Okay, this thing goes on when the complexity just gets worse and worse and worse. Try to think about making a mathematical model that predicts behavior. It's like, are you kidding me? I love this picture. We've talked about these EM. This is not transmission EM, this is EM, but it is scanning EM. The technique here, we'll look a little bit further, is a quick freeze deep etch. You take the rat and you stick its head in liquid nitrogen. Just 4 degrees. 4 degrees Kelvin I'm talking about. Yeah. Okay. Then you chip out the brain. You hammer it. You got to go through the frozen skull. You can smack it and crack it, actually. You can do it like that. Just smack it on the, and it just shatters. Okay. This is a liquid nitrogen temperature. So then you just take a piece of the brain and then you apply a plate on either side of it at liquid nitrogen temperature, and then you pull the plate apart, okay? And just you crack the brain, okay? And it cracks, not surprisingly, along bilayer planes. The weakest point in this whole system is the bilayer. The monophase, each monophase just comes apart. It's just van der Waals interactions instead of all the electrostatic or covalent binding. You don't have a lot of covalent binding. You've got lipid bilayers that are adhering by van der Waals forces. That's the weak spot. So when you crack it, that's what cracks, okay? And then you get to look in on a synaptic terminal with its frozen in place synaptic vesicles, now 3-dimensional structure, and you begin to see some of the complexity that they're not just floating in the synaptic terminal, but they are on some structure, some organized skeleton, and they are going to be moved around the synaptic terminal through the skeleton. Here you can see inside a synaptic vesicle, okay? Here you see the synaptic structure. Here you see the internal face of the receptor, okay, of the postsynaptic junction, okay? Just the bilayer, of course. These things are proteins in the middle of the bilayer, which you'll see on a picture how this happens. just more pictures. I just chose this because it's at a high enough magnification. It's probably about 100,000 magnification. This is a quarter of a micron. This bar is a quarter of a micron. That's 250 nanometers right there. Okay, so these are 250 angstroms, or 25 nanometers, about a tenth the size of that. You can see the bilayer. Okay, so that's why that's there. Here's a good example of, just like we saw a single dendritic spine with 9 synapses, here we see a single synaptic terminal making contacts with 1, 2, 3, at least 4 dendrites. This guy, here's another guy coming in. So again, the complexity of the system. Again, another high mag view. This one I like because you could see all of the postsynaptic machinery that we're going to have to be thinking about. These are all receptors, and these receptors have to be assembled. Not only that they have to do their thing when they're receptor activated by the neurotransmitter released into the synaptic cleft, but they have to be there in the first place. They have to interact. These are not just isolated. When we talk about receptors, you have to get rid of the idea that these are isolated proteins that are somehow floating on the membrane waiting. They're not. They're part of a whole structure that's three-dimensional. You have to modify that. Some of these neurotransmitters are going to modify it by metabodropic receptors. It's not just about ion channels, right? You got all sorts of GTP activated systems that are AMP, cyclic AMP activated systems. All of them have to be next to one another to interact with one another. This is what the postsynaptic side looks like. And look at all this stuff coming out here. Some of these are going to be, if the activity is intense, okay, they're going to the nucleus, and they're going, I'm getting a lot of lip here. Okay, it's time to make a bigger unit. Okay, we've got, this has been busy here. It's time to modify the whole neuronal structure to handle this kind of activity. Again, I love the regularity of this particular synapse and the postsynaptic densities. Okay, and again, the magnification. These are high mag. Just look at the postsynaptic density. Here's your mitochondria. Always mitochondria. Okay, here are a bunch of synaptic vesicles floating around. Here, the density of the synaptic vesicles is high. Now, here's a question. Given the size of this synaptic vesicle, small or large, however you want to think about it, and here's the synaptic cleft. If this guy's going to fuse with the presynaptic membrane, what are you going to do with all these lipids? I mean, what's going on here? Yeah, it's just going to diffuse. If it's going to diffuse, you put 10 of these in that postsynaptic, in the presynaptic terminal membrane, and put a bunch of those in there, and there's a whole lot too much membrane for this. So, I need some explanation here. Okay, so that's what we're doing. The synaptic vesicles now, those 250 are 250 angstrom, are lipid bilayer vesicles. They have a lot of protein on them. We'll see in a minute. They're filled with small neurotransmitters. Okay, they are round. They are clear center. They are close to the membrane surface or clustered typically. That's how you tell the small neurotransmitter vesicles. The neuropeptide vesicles are totally different. They're different size, and they're dense, and they're oval. They're not round, and I'll show you pictures of them. They're straightforward to separate in the EM. The membrane proteins then are to do all these things that I've been going on about. Of being ready, of getting ready, of moving, of docking, of fusing, of coming back out again, of releasing the neurotransmitter, of and stuff we can't even guess about. I don't know why they colored this picture, but at any rate they did. So this is just, I think, a lovely picture of a postsynaptic density. I don't know if it's giving these off, but it looks to me like it is, or it's connected to some array of structure underneath it. And these are what are called dense core vesicles for obvious reasons. And they're much bigger than these. And these are your neuropeptides vesicles. And these are your small neurotransmitter. And you'll see both of them in the same terminal. And that's what we've been saying, and we realized that decades ago. Where are the synaptic vesicles synthesized? Okay, so they're synthesized in the cell body. They're part of the rough ER. Just like all the other membranes, every other membrane that we talked about was synthesized in the endoplasmic reticulum, so are these. And if they're going to put glycogen or glycoproteins or glycolipids or whatever you're going to put, they're going to have to do it in the Golgi apparatus, same as any other membrane associated with any other cell in the mammalian body. Okay, and that's where they are. Well, now you have another problem. All right, the synaptic vesicle is synthesized up in the spinal cord and is used down in the foot, okay, or is synthesized up in the cerebral cortex and is used down in the caudal spinal cord. So now you've got to get the vesicle down there. Well, we already talked about that, right? We talked about fast axonal transport. So you put the synaptic vesicle on the fast axonal transport system, send it down at 400 millimeters a day, and it doesn't take long before you can supply the distal terminal. Well, all right, think about this. Then you've got the synaptic vesicle, which is bundled, packaged, synthesized, cost a fortune to create, right? And you send it 2 days away, okay, you know, 1.5 meters down to where it's going to be used, okay? You don't want to bring it back in order to fix it or repair it or refill it, okay? So then part of the machinery in the synapse has to be to reutilize the synaptic vesicle. Makes no sense otherwise. You can't keep sending this. It's way too expensive. So you've got to reutilize the system. More machinery that has to be present in the synaptic bouton. Okay? This is what a synaptic vesicle begins to look like. And like I said, here we've named about 20 proteins. There are 1,000 proteins that are being used in the synaptic terminal. Okay, this looks complicated. Squat, ain't nothing compared to what's really going on. Okay, but I think it gives you a good sense that you're not just dealing with naked bilayers. So we wouldn't want you to have that idea that somehow we're bringing these naked bilayer vesicles to that. That was my mistake at the beginning of my PhD. I assumed that we were. Okay, that's not true. And as a result, the physical methods that I was trying to use to work on these bilayer vesicles was nonsense because they're inhomogeneous. Okay, my problem. So then here's just a schematic diagram of the nucleus saying, okay, make these guys, Golgi apparatus, axonal transport, reutilization. Okay, that's the basic idea, both for the small neurotransmitters and for the neuropeptides. Neurotransmitters can be resynthesized in the synaptic terminal. Neuropeptides not. Okay, so you're either going to have to reutilize the neuropeptide or maybe have some RNA that's down in the terminal. Oh, wow, that's a whole nother level of complexity. So you're actually going to do some translation down in the terminal. Then you're going to have to regulate it. Yeah. Worse and worse, more and more complicated. Not worse and worse, just more and more complicated. Okay. When you start to now look at now I want to get into the synaptic range. We'll leave the vesicle, we accept that it's there, and we look at how we get to fusion. We're going to start with the idea that when you look down on the synapse, you're looking at a hexagonal grid, that the synaptic vesicles are then brought to the active zone by a set of machinery, which we saw on that quick freeze deep etch photo. We've got lots of dense projections between them, and that's what we're saying. So here you go. This is just a diagram of the freeze fracture technique as I explained it to you, where this is your bilayer, and this is frozen with a plate on one side, this is frozen with a plate on another side. I pull them apart, they crack along the bilayer, and now I'm looking at the intramembrane. This is not even a surface. This is stuff that's stuck in the bilayer as I broke the bilayer in two. And that's what you look at when you look at quick freeze deep etch. The reason that you can see it is because then you spray gold all over it, molecular gold onto it. And that, of course, is dense in the scanning electron microscope. So what you're actually looking at is a gold painted image at the molecular level. This is your hexagonal array. This comes from a neuromuscular junction, And you can see how this is, again, a quick freeze deep etch. And you can see this is the unstimulated receptor end plate of a neuromuscular junction. And here it is after it's been stimulated. And you can see all of the holes where synaptic vesicles are now in contact and have released neurotransmitter into it. And you can see that there is some redundancy now to the membrane. There's too much material in this membrane, and it is definitely becomes unstable. This is just sort of the hexagonal grid that this thing is meant to represent. And this is, again, to show you the molecular scenery and the internal grid. Another one here, this picture just happens to catch some terminals as they're releasing their contents into the dendritic space. And so that is a synaptic cleft that we're looking at here. We're actually inside the synaptic cleft. We already said, or we didn't, the synaptic cleft is 20 nanometers wide. The synaptic vesicle is 25 nanometers. It's too big, so the vesicles do not enter the synapse. Okay, it's too big to go into the synapse. All they do is fuse, they appear to fuse with the presynaptic membrane. They release an incredible concentration of neurotransmitter and ATP into the synaptic cleft. And 5 millimoles is that's a lot. 5 millimolar anything is a lot. I mean, that's as much glucose as there is right in our serum. That's a lot of neurotransmitter. So it's extremely concentrated in this very, very small space. It is removed in about 2 milliseconds. And there are various ways to remove it that you're going to study as you study each neurotransmitter. It could be by uptake into the astrocytes, which are bounding the synapse. It could be by acetylcholinesterase, which simply destroys the neurotransmitter, that sort of thing. Again, I say the synaptic cleft is not empty. There are, as I tried to show you, pillar-shaped protein complexes that make vertical lines across it. And there are all kinds of proteins within the synaptic cleft as well as on the postsynaptic membrane. And now just to reiterate some of these pictures that I saw, and this is one with the vertical poles that are going across. And again, ubiquitously, the mitochondria always in communication. You've seen some of these pictures. I'm just showing you again now that we've done some talking. These are higher mag. This is a synaptic cleft. This is a bilayer. This is a bilayer. This is a synaptic cleft. This, you can see the vertical lines going across the synaptic cleft. The magnification here is probably at about 250,000 times. Okay, these are, you don't get these pictures very often. Okay, I mean, you can work for 6 months before you get a stable piece of tissue that will allow you, the electrons that you're throwing through that thing are very complicated. And so they usually pop when you try to see them at this magnification. These are gorgeous pictures. This is a 100 nanometer bar here. I mean, these are great skill to create these images. And so you can see it, again, look at how far the structure extends into the postsynaptic cell. Okay, look at the bilayers, look at the proteins on the bilayers, look at the neurochemistry, these macromolecular neurochemistry lectures that we've been talking about really, in my mind, sit at the interface between electron microscopy and chemistry. Okay, there's no real boundary here. We're now moving into the world of molecules. Okay, you're looking literally at molecules here. On the postsynaptic membrane, you've already seen these. You have all these densities that extend into the cytoplasm, which I just showed you. You can see that there are clusters of receptors of associated proteins. Again, just another picture illustrating what we've been saying. Here you can see, we're going to get to this in a minute, but I want you to see the postsynaptic densities. Again, pictures, all of them out of this book. Here's membrane that appear to be extending off into the dendrite. I think there's an enormous amount that we don't know yet about this. Despite all these years, what have we been doing? This 50 years. Next year will be 50 years on the report of Whitaker and DeRobertis, 2 separate labs, on the isolation of synaptic terminals. That's going to be a 50-year anniversary, and there's so much we don't know. It's amazing, and so much that we're learning, which we're going to get to very shortly. Okay, let's talk about the problem of synaptic vesicle recycling. The neurotransmitters are actively transported into a synaptic vesicle. As we'll get to in a minute, the main gradient that the synaptic vesicle makes is an acid gradient, and it consumes the acid gradient. It acidifies itself in relative to the rest of the synaptic terminal, and then it uses that acidification to refill itself with neurotransmitter. That's what it uses to power the reuptake of neurotransmitter back into the synaptic vesicle. Acidification is very fast. In case you don't know what the fastest enzyme in the known world of enzymes is carbonic anhydrase, okay, which has a turnover rate of 32 million per second. Okay, that's carbonic anhydrase. That's the fastest. So if you want a fast enzyme you want to pull the neurotransmitter, you got a lot of activity going on. Okay, you're trying to recycle with a 2 millisecond half-life. Okay, you have intense activity coming in. You need to refill your vesicles that are being consumed, and you don't want to exhaust the whole system. The fastest gradient you can create is in this acid gradient. Okay, everything you need is sitting there. Okay, all you need is carbon dioxide. Okay, carbonic anhydrase, acidify, use that, fill yourself back up, go back to work. Okay. You also have the synaptic vesicles, which, as we saw on the EM already, cluster in front of the active zone and dock with the terminal plasma membrane. That these vesicles are primed. They're primed for membrane fusion. The vesicles that are floating out in the synaptic terminal, look like they're floating, cannot fuse with the membrane. They are not primed. They are not ready to go. That's a separate step. And after priming, the entry of calcium then leads to fusion and pore opening. And then there are lots of options for recycling, which we need to look at all this before we stop today. So this is just a diagram of, we're going to look at a couple of cartoons here. Here are the synaptic vesicles coming off the axonal transport system. They are now available for this cycle. Here they are, neurotransmitter, moving toward the active zone, becoming primed, docking, entry of calcium, fusion, release of the neurotransmitter, right? Activation of the receptor, passing ionic transport across through the receptors, okay, through the ion channels, okay? Recycling of the membrane back out, in this case, using a Clathrin cage. That's not the only way to do it, which we'll talk about in a minute. Here's a more detailed picture. And this is the beginning of the cycle right here. So you have the transmitter using a proton pump, creates an acid environment, refills itself with neurotransmitter. This then stays via actin and synaptins. Now is sitting on close to the edge. It's on a structure, but it's not really ready to be used. Before it can be used, it has to go into a mobilization step, which uses a calcium calmodulin kinase and other synapsin entered this structure, which I think you saw beautifully on that quick freeze deep etch photo, okay? Then you move it into a docking stage. Each of these is named. The docking stage uses GTP binding proteins and snares, which we're going to look at in detail in a minute. And then these are your snares. They bring the synaptic vesicle in contact with the presynaptic terminal membrane. That's a good term for it, presynaptic terminal membrane. And now bring it adjacent, and then with the entry of calcium is a fusion step which releases, okay? Here at the priming stage, you have a ton of enzymes now which are involved, and obviously the fusion also with synaptotagmin, which we'll talk about, snares, complexin, I mean, all these things that DOC2, I love it. You know. All right. They are actively transported. The synapsin is reversibly bound to these synaptic vesicles. It holds the vesicle in the reserve pool. We've already talked about the enzymes, so we can move on and reiterate that. The synaptic vesicles cluster in front of the exact active zone, and then they dock with the terminal plasma membrane. These are going to use GTP binding proteins and snares. Snares. Snares, the term snare has no real good meaning, except that it's cute to call it a snare because it grabs the vesicle and holds it in place. It stands for synapse N-ethylmaleimide. You don't care what it is. It's a technique that's used for isolating these vesicles. It doesn't matter. Okay. Here's synaptobrevin. Okay. Here's synaptotagmin. Okay. And here's SNAP25 and syntaxin. These names, there's so many. It's unless you're going to do research in the field, I don't see any point in learning them, but you will hear these names. The thing about the system, and what I find rather strange, honestly, is that this whole molecular system right here is insensitive to calcium. It won't react with calcium. Calcium cannot trigger this system of synaptobrevin, synantaxin, and SNAP25. And this is your machinery that's going to cause the fusion. And you go, how could calcium is the signal? You can't, wait. Okay. So in order to make it sensitive to calcium, why they could not have calcium be part of their system, I don't know. Okay. But in order to make it calcium sensitive, which it must be, you have this synaptotagmin, whose only function is to impart calcium sensitivity to the function of this complex. Why the brain evolved that way, why the synapse evolved that way, I have no idea. And the secret is in evolution, I believe. They're then primed to convert to a state of competence, and then you have now these ATPase. This is where the term SNAP comes from. Like I said, this is just technique issues. It's not relevant. And we've already talked about all of this. And this is a nice three-dimensional picture of the synaptobrevin coming on off. This is now your vesicle, sorry. Here's your vesicle. Here's the presynaptic terminal membrane. And here are, they're now in intimate contact with one another, right? But have not yet fused. We'll come to that in a minute. But they are ready to fuse now. You have all the machinery. You have the synaptotagmin, which makes it calcium sensitive. You have synaptobrevin, you have the SNAP25, and the syntaxin. Okay, and we've said this. And then you add in another protein called complexin, which, so what the complexin is going to do. So if you have all these proteins, let's go back a picture. This protein complex, right, is, if you leave it where it is, then how are you going to get this to release this when this is in the way? In order to cause the lipid bilayers to fuse, you're going to have to get rid of the proteins that have caused all this docking and priming to occur. That's what complexin does. The complexin comes in and it takes the protein out of the way. But if you want to know the real truth, I don't think anybody knows how this happens yet. This is all a fairy tale. From the next several slides is like my imagination, somebody else's imagination. We're all drawing pictures and we have no idea what's going on, okay, at the molecular level, because it just doesn't make sense, because you know from prior lecture that you can't flip-flop lipids. You can't take a phospholipid from one side, and I searched this last night for this talk. You can't, you do not find evidence that phospholipids from the synaptic vesicle are showing up in the terminal membrane, okay? But if you're just literally fusing them, okay, then and you're fusing, by the way, you're fusing them in the wrong way, because this is the outside and this is the inside. So you're fusing an outside monolayer with an inside monolayer. No, you can't do that, right? The whole synthesis was, and then you're going to reuse the thing, right? You can't. The idea of it. So nobody has constructed a model, a molecular model, which allows this fusion to occur without mixing lipids. And yet you know that you can't mix lipids. It won't. The system won't work. The vesicle will not function if you're going to do that. So that doesn't work. At any rate, what this is, and you have to picture this in 3 dimensions, is that you then somehow insert this pair of scissors, one end of the scissor into the vesicle, one end of the scissor into the postsynaptic, what do we call it, the presynaptic terminal membrane. And then you take the scissors and somehow snip the phospholipids to pull the proteins and phospholipids out of the way so that the vesicle is in communication through, that's the key, through the presynaptic terminal membrane because the contents have to exit fast. Millisecond range, it's got to dump 5 millimoles of neurotransmitter into the synaptic cleft. So this is the cartoon that is drawn. It doesn't work. That's all I can tell you. It doesn't work. This cannot be, I get it. That's like, I like the picture. It doesn't work. Okay? And yet here they are. So we're missing something. Okay, there's definitely something that we're missing. Now, I like this drawing a lot. So I don't like this drawing. It's from the same book. Look at this, okay? So here's this is a nice illustration of a step-by-step what happens at the active zone. So already, all of us are prime, the calcium is already coming. We're in the active zone, and you somehow blob out all the contents into the synaptic cleft. That's really cute. I like it. That's a riot. I don't believe it, okay? I like this one. Look at this one. This has a different model, really. This takes proteins and assembles the proteins and then says, I'm not going to mix the lipids, but I am going to move the proteins of the synaptic vesicle to make a big, wide pore in the membrane. And I'm going to fuse that pore, like the docking, just like Eagle docks with, who is it? It's Eagle and Columbia, is it? That Eagle docks with Columbia, the lunar orbiter docks with the main ship and comes back, and you don't fuse the two of them. You create a lock between them, a collar. It's a collar, and so the idea that this creates half of the collar and then docks onto another half collar, and then you open the hatch, and then you close the hatch, and then you undock the collar. I think that makes much more sense. And that's what's displayed in this diagram, because this eliminates the problem with the lipids, which I don't get. Anyway, then you have to undock it and reuse it. There are 3 cute terms that are used. You can kiss and stay, kiss and run, or be recycled via clathrin-coated pits. Kiss and stay means you drop you dock, you drop your neurotransmitter, you undock, not undock, but you close. Let's close the pore or whatever it is. You unfuse, whichever is occurring. You stay right where you are, acidify, pick up neurotransmitter, and do it again. Just stay where you are and keep doing that, okay? Of course, you're doing this 500 times a second, okay? But that's what we're talking about. That's kiss and stay. Kiss and run means you drop your packet, and then you are recycled through the system, okay? Clathrin pit is clathrin pit. So I'll show you. So this is the kiss and stay. So see, there's no recycling. Kiss and run, okay? And then this one, you go all the way through, you go back into the endosome, which we haven't been talking about much. You rebut off, you remake this synaptic vesicle. Maybe this is maybe this is part of the answer if you pick up too many lipids. I don't know. Maybe that's why that's there. I don't know the answer. This is the diagram. Don't you love this picture? Doesn't it look like one of those toys that you get to collapse and not collapse? They sell these in the toy stores if you have kids or grandkids. And notice that they show that actin, remember that was remember where actin was? The actin bundles were the cytoskeleton that underlie the plasma membrane. Those are functions of near the plasma membrane. They need to get the heck out of the way to make room for somebody else. You're not going to wait for them to just float away. They don't float away fast enough. So you're going to put them on the train and pull them the hell out of the way and get out of the way. I need more space. And this is Captain Crunch, sort of at high mag, right? These are your clathrin-coated pits. Okay, so they're real. I just love the honeycomb, right? I love it. We've talked about this acidification. We've talked about this. We've talked about all this stuff now. This is, again, just a diagram of some of the complexes that occur. Don't think that this acetylcholine, you can't even pack. If you tried to fill the synaptic vesicle with acetylcholine, just concentrate it as mad and just fill it with acetylcholine, you couldn't get as much acetylcholine as in that thing. Okay, it's super concentrated because it's complex, okay, in a way that it wouldn't, if you didn't complex it with something, it would not allow itself to be packed that densely. So these things, and it's ATP is what you're using to pack it, which is sort of an interesting system. And then you release the ATP and you have all the energy that you could possibly want at the same time. And this is just talking about things that we've already talked about, really. We're not going to get into this. This is last year's lecture, calcium entry into the cell. Neurotransmitters, the neuropeptides, the only comment that I'll make at this level before we have a talk later on in the year on neuropeptides, the only thing I would say is that you, generally speaking, the dense core vesicles never come into the active zone. They do not fuse with the presynaptic terminal membrane that we have been talking about. They fuse out here on other parts, on non-synaptic terminal portions of the synaptic terminal. Now, if some of you may remember from last year, this ought to ring a bell when you see this happening. So, for instance, if I asked, where are the NMDA receptors? Not the glutamatergic, not the metabotropic, not the AMPA terminals, but the NMDA terminals. Where are they? Do you find them in the synaptic cleft? No. They're not, that's not where they are. They are out here. And they, as you may remember, are only activated when the calcium concentration of the synaptic terminal is very elevated and sustained. And when they're very elevated and sustained is when the neuropeptides are also released. And that's because both the NMDA, I mean, that's learning, right? If you activate the NMDA channel, then the postsynaptic cell is going to be learning. You are going to be modifying. The nucleus is going to be activated by this activity. And that's true for the neuropeptides as well. We're really done, and that was the terminal. I mean, you're going to get into terminal activation, so we're done, okay? But you need to walk away from this lecture just going, I'm never going to understand this. Okay, none of us are ever going to understand this. This is infinitely complicated. If science were to be able to continue at the current pace for 50,000 years, we're not going to understand the pictures that I just showed you. Yeah. Yeah, I mean, well, I think here's my rule. This is, people ask, is it going to be on the exam? I used to run the neuroscience course for the medical school, and everybody, all the medical students know, what's going to be on the exam? And here's what I say. Here's my answer to them. If I say it once during a lecture, it's not going to be on the exam. If I use the word more than once, it's fair game on the exam. So there weren't a lot of, so the concepts that I've laid out, you might see those concepts. Where are synaptic vesicles synthesized? And synaptobrevin, synaptotagmin, these named as snare. Where do you find a snare protein? I mean, I don't know what they ask on the boards. I don't, but if I haven't, it would be stupid of someone writing the boards to put on the boards something that we glance over in order to help you understand the concept that we don't use otherwise. That would be dumb. Yeah, I mean, you know, you could see a picture of a linear array like that and look at it and go, that is not a cerebellar synapse. You know, that linear array is neuromuscular. You know, that's motor end pipe. I get it. That's the sort of thing that they might do. But anything that would be on the boards is, you know, they're going to get 1 to 2 questions on this lecture. Then you're not going to be talking about the kind of detail that we're talking about. But this course is not about the boards. This course is about helping you to appreciate what you're doing and understanding. This is, you really